Welcome to the North Star ALS podcast, where ALS science meets ALS families. I'm Nadia Sethi, co-founder and CEO of North Star ALS. Our mission is to make ALS research more understandable, more accessible, and more connected to the people it ultimately serves. We offer support for ALS families to connect with research opportunities, help contextualize current research findings, and bring insights from ALS families directly back to researchers. Today, we're focusing on biomarkers in ALS and their role in more targeted drug development in ALS. I'm honored to be joined by Dr. Michael Benatar, Professor of Neurology and Public Health Sciences at the University of Miami, Executive Director of the University of Miami ALS Center, and the Walter Bradley Chair in ALS Research. Dr. Benatar is an internationally recognized clinician scientist whose work has helped shape the modern landscape of ALS research. He leads the groundbreaking pre-symptomatic familial ALS, or pre-FAL study, which has transformed our understanding of the earliest stages of inherited ALS, and he serves as principal investigator of the CReATe Consortium, advancing research across ALS and related neurodegenerative diseases. Dr. Benatar, thank you so much for joining us. It's an honor to have the opportunity to learn from your perspective on how our understanding of ALS has evolved and how that knowledge is guiding the next generation of therapeutics. To begin, we've often described ALS not as a single disease, but as many diseases that can look similar. Can you explain what that means and why recognizing heterogeneity is so important?
Yeah, perhaps I can first say just thanks for the kind introduction and a pleasure to join you on this podcast. And always a pleasure to have an opportunity to talk with you and directly to our patient community. So I think the idea of ALS being many diseases has sort of many facets to it. So maybe we can try to sort of tease it apart a little. The first is the idea that there can be many different causes of ALS. That's one way in which there's heterogeneity or variability. And we know there are different causes from what we've learned in genetics. There are people who have a mutation in SOD1, for example, or C9orf72, or TAR-DBP. These are different genetic mutations in different genes that all lead to ALS. So these are genetically or etiologically, fancy word for saying the cause of disease, where the disease is different. Then there's, and I'm going to leapfrog over biology and come back to it, but the disease is very varied phenotypically in terms of how it presents, or not so much how it presents, but how it runs its course. So there are people from first symptoms to the end of disease that can go for 6 months. There are people from first symptoms to the end of disease can run for 30 years. So the disease is very heterogeneous phenotypically. The in-between piece is the biology. So what's driving how the disease progresses and the speed of it? I don't think we fully understand that. What we do know is that most ALS, about 95, 97% of the numbers that are in the literature, is associated with a particular pathology in the brain called TDP-43. And I'm sure we'll talk more about that during the course of this podcast. But there are people, for example, people with SOD1 ALS, who, as best as we currently understand, don't have TDP-43 pathology. Instead, they have SOD1 pathology in the brain. So if you contrast, for example, people with SOD1 ALS to, say, people with C9orf72 ALS or non-genetic ALS, the cause is different, the biology is different, and the speed with which disease progresses can be very different. So there are all these sources of heterogeneity or variability, but what makes them look quote unquote alike or similar is that the final common pathway of what's affected in everybody is the motor neuron system. And there's progressive degeneration and loss of motor neurons that leads to progressive weakness in the arms, the legs, the bulbar region, the breathing muscles. And so from the outside, sort of, or even from the inside, how the disease feels, what it affects, what we observe as clinicians, it looks the same.
Thank you so much. That was a great introduction to what we're going to be talking about, which is biomarkers. So maybe you can we can take a step back and talk about what is a biomarker. I think it's in some ways it's really simple and in some ways it's really hard to understand. So maybe you can give us just a brief overview of how biomarkers, what biomarkers are, and then we'll circle back to how that plays into heterogeneity.
Sure, happy to. So I'll try to give a definition, although the definition is sometimes a bit overly sort of complex and contorted, and maybe examples are sort of easier. So maybe we'll try to define it both ways. Um, as a definition, a biomarker is something that we can measure, typically in a biofluid or physiologically or imaging or in tissue. So it's something that we can measure that tells us about underlying biology, physiology, or sort of pathobiology. What I mean by pathobiology is biology that's become disease, that's abnormal. A good way to think about this from our recent collective history is a COVID-19 test. You know, you have symptoms, you go and get a swab, you get a PCR, you get your COVID test result. That's a biomarker. For anybody who's ever gone to have their cholesterol tested, that's a biomarker. For anybody who's ever had a pregnancy test, that's a biomarker. These are things that we can measure that tell us, for example, with COVID, have you been exposed? Do you have an active infection? Cholesterol, are you at risk for a heart attack or a stroke? A pregnancy test, are you pregnant? And so these are the sorts of examples where I think people are really familiar with these on an everyday basis, but maybe don't think about them as biomarkers. So it's a complicated idea, but I think everybody's probably super familiar with them in sort of day-to-day life.
Yeah, that definitely helps to give things context. And can you tell us about the different types of biomarkers? And I think that's where it starts getting a little more complicated.
Yeah, it does indeed. So I think there are different ways to sort of pass what we mean by different types of biomarkers. I'm going to allude to one, but then not spend most time on it because we touched on it already. One way to think about a biomarker is what's the source? Is it in a biofluid like blood, urine, spinal fluid? Is it an MRI? Is it an imaging, characteristic? Is it an EMG, a physiological measure? Or is it a tissue measure? Is it a biopsy? Is it a skin biopsy? Or in the cancer world, is it a biopsy of the cancer? All of these are sort of sources of biomarkers, but none of that tells us what type of biomarker something is. So when we think about types of biomarkers, we have to really think about what do we want the biomarker to do? Um, Let me try to explain what I mean. So if I'm a neurologist and somebody comes in, not if I'm a neurologist, I am a neurologist. Somebody comes into clinic and they've had weakness for the last 6 months, and we're wondering, is this ALS? We're uncertain if it's ALS or, for example, it's myasthenia gravis or a different neurological disorder. Imagine I had a blood test that I could send to the lab and it would tell me you have ALS or you have something else. That would be a diagnostic biomarker. That's differentiating people who have the disease of interest, in this case ALS, from other diseases or non-diseases. I'll just say right there that we currently don't have any diagnostic biomarkers for ALS, and maybe we'll talk more about why that is and how helpful they would or wouldn't be. But a diagnostic biomarker is one kind of biomarker. A very different kind of biomarker is a prognostic biomarker. So to sort of make clear what that term means, um, when you go to your doctor and they tell you you've got some condition, let's use the cancer example. How long do I have to live with cancer? Patients with ALS may ask the same question. What we're asking about in that sort of scenario is what does the future hold? And a prognostic biomarker is one that gives us that prognostic information. What that means is it tells us something about the future. That may be the future rate of disease progression. It may be how long you will live before, for example, needing breathing support or ventilatory support. But in essence, a prognostic biomarker tells us what the future holds. That's really, really important information for anybody facing this disease, but it's also really, really important information when we're doing drug development and clinical trials, and perhaps we'll come back to that. Another kind of biomarker is what we call a disease progression marker. This is a marker that changes as disease progresses. So as disease gets worse and people become more disabled, the level of the biomarker goes up or goes down, but it's changing over time. Another kind of marker that we can measure longitudinally is something that we might call, and this is not FDA terminology, this is mine, a temporally stable biomarker. Think about a biomarker that is, say, elevated in people with ALS and stays elevated, but doesn't change over time. So it's not changing, it doesn't track disease progression, but it is elevated and abnormally so. And there would be value in an experimental clinical trial of lowering that biomarker and returning it back to a normal level. That would be helpful. So having a biomarker that's elevated but stable over time, and I'm sure we'll talk about neurofilament light chain today, but NFL or neurofilament light is an example of a temporary stable biomarker. It's elevated early in disease and then remains relatively stable. And if we can bring it down with the therapeutic, that's a good thing. But now I've introduced the idea of a response biomarker. A response biomarker is one that literally responds to or changes when we give an experimental drug. So if the biomarker returns to normal levels or heads in the direction of normality, that's a good response. That can tell us that the drug is doing something. We don't yet know whether that's going to benefit the person in terms of function and how they feel or how long they live, but at least that biomarker can tell us that there's been a biological response. Here, the analogy to cholesterol is helpful too. Imagine you're testing your cholesterol and finding it's high because that carries a future risk of a heart attack. We know that if we can lower your cholesterol, we can lower the risk of a heart attack. We don't want to wait till you have a heart attack to measure it again. We want to measure it when we give you a drug and show that the levels come down, and that's helpful. And if we have those sorts of response biomarkers in ALS, they can tell us when a drug or an experimental therapy is doing something. There are other kinds of biomarkers, but these are the ones I think we talk most commonly about sort of in the ALS world. And so I'll pause there.
Thank you so much for that. Yeah, I think with the cholesterol example, I've heard it described that, we don't really care about our cholesterol. We care because our doctors tell us it matters. Um,
I think that's right. You don't feel a high cholesterol,
yeah.
but you feel it when you have a stroke or you have a heart attack, and we don't want to wait for that.
Yes. Yes, exactly. Okay, so we talked about lots of different types of biomarkers. We'll come back to some of those. Can you talk a little bit about why it's so hard to identify biomarkers and what it means to validate a biomarker?
Yeah, gosh, there's a lot there.
Yeah.
So let's sort of talk first about development. I guess if we think about development of a biomarker, there's a spectrum along, or there are multiple steps along a spectrum. So first we have to discover something. We have to identify something new or something that we think could be sort of repurposed for a biomarker that would be useful to ALS. So there's sort of a discovery phase. And very often that's in the laboratory. Somebody's got an idea of something to measure. They've developed a new antibody that can measure something. And so there's that sort of discovery piece of the essential reagents and the idea that are necessary to begin the development. And sometimes that may be done in tissue, post-mortem tissue. It might be done in a cell or a culture model. There are all sorts of ways in which that discovery can happen. But at some point in the discovery, people then want to test this in a way that's more relevant to people. So they reach for the biological samples that are in the freezer, the blood that was collected, the CSF that was collected that's frozen, and they measure that biomarker in those samples of convenience that they have available. It's at that point that I think it really matters that somebody needs to start thinking about what's the purpose of this biomarker? What do we want it to do? Do we want it to be a diagnostic? Do we want it to be a prognostic? Do we want it to be a response marker? Because thinking about those things early tells you, as you fine tune the assay, get it working really well in the laboratory, What's the next set of clinical samples that you need to test the biomarker in to test that hypothesis that it might be useful for the intended purpose? So before we get to actually doing that larger experiment with human samples, there's a lot of work to be done to make the assay robust. We sometimes call this analytic validation. So I'm breaking up the word validation into different parts. So the analytic validation is making sure that the assay is a reliable, robust assay. What does that mean? It means if I measure it today and I measure it tomorrow, I get the same result. If I take the sample and I leave it on the bench at room temperature for 24 hours, do I get the same result? If I take the frozen sample and I thaw it and I freeze it and I thaw it again, do I get the same result? If I buy another lot of reagents from the lab that I'm using, do I get the same result? If I dilute the sample, do I get the same result? All of these things are part of making sure that the test is actually measuring what we want it to measure, and it's doing so in a way that's precise. I'll go back to the cholesterol example, or let's go back to the pregnancy test example. Imagine you had a pregnancy test that today tells you you're pregnant, and next week says you're not, and 2 weeks later says you are. Well, what do you believe? The assay is probably not working. So that test has to measure what we want it to measure. That's part of the sort of analytic validation. Once we've really sort of sorted that out, now we get to the clinical validation piece where you have to say, we're now going to test this robust assay and say, can it do what we want it to do? Can it, for example, if we want it to be a marker of disease progression, as disease worsens longitudinally over time, does the level of the biomarker progressively go up or progressively go down? Or does it just sort of bounce around, in which case it's not going to be a useful biomarker of disease progression? If we want this as a diagnostic marker, can it differentiate people who I think have ALS, but I'm not sure, from people who I'm not sure and turn out to have ALS or turn out not to have ALS? So that's a whole different population, and you might not need to measure it longitudinally. You might need to measure it just once. So thinking about, and I'm going to use this term that the FDA likes, context of use. That's a fancy way of saying, what's the intended purpose of the biomarker? We have to think about what that purpose is so we know how to design that clinical experiment and then, quote unquote, validate the biomarker for that purpose. So I really don't like the term validate because it's such a loaded term and it means different things to different people. So I think maybe it's helpful to think about getting the assay working in the laboratory and then testing it in relevant human samples that inform whether it has the utility that we want to use it for, sort of in the real world.
So, on that same note, I guess, what does it take for the FDA then to recognize a biomarker for a specific context of use?
Excellent question. What it really requires, I think, well, I should give a caveat first. I don't begin to read the mind of the FDA, so I can tell you what I think and know sort of from the outside. So the FDA has something called a qualification program. You can formally qualify. What that means is the FDA gives a stamp of approval. They recognize a biomarker can be used for a particular context of use. Now, when last I looked, the FDA has only ever approved or qualified, I think, 8 biomarkers and none for neurological disease. My own view is that the qualification process is somewhat cumbersome and perhaps unnecessary. So the listeners to this podcast may know that when the FDA gave accelerated approval for tofersen or Qalsody as a treatment for SOD1 ALS, that was based in part on the fact that the drug lowered neurofilament levels, neurofilament light levels, also heavy, and it lowered it in a meaningful way. I think it's a 60% reduction. And the FDA didn't just sort of, in a sense, not make it up. They didn't just sort of suck this out of their thumb and say, well, this looks impressive. There was a rich body of evidence that tells us, one, NFL is elevated in people with ALS. Two, it tends to remain elevated and stable over time. Three, people with higher levels progress more quickly and don't live as long. and so it has prognostic value. And so all of that from many years and many publications, this robust body of evidence and literature, they could turn to and say, in the untreated state, we never see a 60% reduction in NFL. A reduction in NFL will be expected to be reasonably likely to predict a clinical benefit because lower levels are better than higher levels and associated with more slowly progressive disease. And so the FDA showed us that they're willing to incorporate robust evidence from the peer-reviewed literature, even if not formally qualified, to make decisions about drug approval. And that's not to say them, and again, I don't know what the FDA would or wouldn't do, but it's not to say that they would or would not approve or a drug partially based on biomarker data in the future. But what we need to do as an academic community is make sure that we have this robust body of literature that the FDA and other regulators can lean on to support decisions about drug approvals.
I think that, yes, that makes a lot of sense. So if there's a hypothetical drug in existence that had a clinical trial and there's movement in a biomarker, how, I guess the context of use would matter. How do we determine if that's enough? We see a lot of noise about what biomarkers mean and how they matter. So how do we interpret that information, I guess? And yeah, maybe you can.
Yeah, it's an excellent question. I think it's lots and lots of good data. So let's start with neurofilament again, because we have so much information, and even there, our knowledge is incomplete and there are open questions. But one of the things we know is that if you just follow a large cohort of people with neurofilament levels over time, you can see some people the levels go up a little, some people the levels go down a little, some people the levels stay stable. On average, they're mostly stable. They may go up a little bit over time, maybe 1% per month is our sort of best estimate of this, but it's sort of modest. So what that means is if you're going to talk about a 1% lowering of neurofilament over 6 months, I'm going to shrug my shoulders and go, that probably is within sort of biological and analytic noise. If you tell me you reduce NFL by 60%, I'm going to say, well, that's unheard of. We don't see that in the untreated state. Now, there's a lot of range in between that. What's the right number? I don't know that any of us know for sure, but I would say from the published data, something in the 25% range is probably outside the range of what we see. Of course, one has to be careful if you're looking amongst 3 people. Those could be very unusual people. So I'm assuming we have a reasonable sample size and we're looking at sort of an aggregate measure here. But we can sort of begin to make these guesstimates because we have such robust peer-reviewed data. For any new biomarker coming over the horizon, let's say somebody's proposed something as a biomarker that's elevated in ALS versus controls, and they've only ever looked cross-sectionally and not looked longitudinally. And then they say, look, in response to experimental drug X, this biomarker goes down 20%. And oh, it was an open label study. We don't know what happens without the drug. I'm going to go, well, I have no idea whether that means anything because we don't have the evidence to interpret it. So I guess the way I'm trying to answer the question is to say we have to walk before we run. If we want to know if something can tell us about a biological response, 1, we have to have a control group, but 2, we have to know how that biomarker behaves in the absence of giving that experimental drug.
Yeah, that makes a lot of sense. So can you talk a little bit about biological response versus clinical response? So if we move a biomarker one way or the other, and we talked about this a little bit, but how do we know that moving a biomarker one way or the other is actually going to have a real impact on how somebody is living with the disease?
Yeah, it's a great question. I think we're in the early stages of sort of figuring this out. So again, I'll come back to NFL. So we already know that if your NFL level when first measured, let's say enrollment in a trial, is 200 picograms per milliliter, I'm going to forget the exact numbers, but compared to somebody who's got half that, you're progressing 2, 3, 4 times faster. So if we could halve that based on everything we know, halve you from 200 to 100, that's likely to exert a benefit because the way we think about NFL is it's kind of a readout. It's like the speedometer. This is a good analogy. Let me try to answer this question another way. Imagine you're driving and you're driving towards your destination and at 50 miles an hour, it's an hour's drive. And the destination here is the end of disease. It's death from the disease. If you're driving at 50 miles an hour, it's going to take you an hour. Imagine we could slow you to 25 miles an hour, right? Now take 2 hours to get where you're going. So we would prolong your survival because we've slowed the speed of the car. We've slowed the speed with which the disease is progressing. And we have a sense of that because of how much we know about NFL. What we still need to get, and we're beginning to see this from sort of the real world experience of tofersen, is that not only do we lower neurofilament, but we prolong survival, we slow the rate of functional decline. People are retaining function, living longer than we would expect based on everything we know about SOD1 ALS. And so we're beginning to sort of build that body of literature and evidence that substantiates what we suspected based on first principles. Now, if you come up with a novel biomarker, we're going to have no idea yet what that means in terms of translating into something that has functional meaning for somebody with disease. And that's a really important knowledge gap that we're only going to close through research.
That's really helpful to see. And what what types of research would help us understand that gap? How would we understand if a biomarker has a clinical effect in somebody? How do we pair that data together?
So again, I think it's multi-step. So we learned about the potential utility of NFL from natural history studies. Just following people over time with careful phenotypic data, understanding their function, and then measuring neurofilament levels over time. That can only ready a biomarker to serve as a response biomarker. Ultimately, you need an experimental therapeutic. You need to intervene and show that you impact and lower that biomarker. So it's multi-step. If you go back to that paradigm we laid out of development, discovery, analytic validation, figuring out context of use, measuring in a clinical cohort that tells you, for example, the biomarker is stable over time. And then the next step is introducing this into clinical trials and trying to sort of base regulatory decisions and decision making in trials based on that biomarker data. So again, you have to walk before you run. You need all of that information along the way.
That's really helpful to hear. So, can you tell us a little bit more about the value of observational studies and longitudinal studies? We hear a lot in the patient community that people want to try interventions, and it's very understandable given the nature of ALS. But those interventional trials don't always give us information about the disease and about biomarkers. So, can you maybe go into that a little bit, the value of the observational studies and how they make a difference in biomarker development?
Yeah, so maybe just first to acknowledge that I totally understand and agree that the prospect of participating in an interventional study with the hope for a benefit, but recognizing that every clinical trial is an experiment. You could get worse, you could get better, it might have no effect. That makes sense. Not everybody gets to participate in a clinical trial. Either they're at a place where a clinical trial is ongoing, or they're not eligible, or they choose not to for whatever reason. And observational studies, especially those that are designed to help us discover, develop, validate, using a word I don't like, but figure out the context of use for a potential biomarker. All of that is essential to help us conduct future clinical trials in a way that's more efficient and more effective and more likely to yield clear answers. Here it's worth pausing to say that For decades, the ALS community, and this is not a criticism, it's just an acknowledgement looking back over time. For decades, we've done clinical trials that have been quote unquote negative. They haven't yielded a therapeutic benefit for patients. And we've learned precious little from them because there weren't biomarker studies embedded in them, or the biomarkers that were, we didn't sufficiently well understand. And so we don't learn things, for example, like did the drug get to where it was supposed to go? Did it get into the central nervous system? Did it get into the motor cortex or the spinal cord? Did it engage its target and have the intended effect? Did it affect the biology in some way? If we have a drug that lowers neurofilament but doesn't help people, that would be super important information, right? So every study that we do, whether it's interventional or observational, can add to our body of knowledge about biomarkers. And in turn, that can help make the next clinical trial or set of clinical trials more informative and more likely to give us a clear answer. Help us triage out the drugs that are likely not going to work and double down on the drugs that seem to have promise.
Um, Thank you so much for that. And do you see limitations in our current data sets? I guess what I'm trying to ask is sometimes we may have data sets that may be skewed towards one genetic subtype. How might that skew research findings? How do we overcome things like that? I think with our biobanks too, I feel like there may be limitations in the types of samples that are there, and the types of ALS that's represented.
Yeah, it's a fair question. I mean, I'll say in general, observational studies tend to enrich for people with more slowly progressive disease. And I think that's a natural phenomenon. If somebody's disease is progressing very quickly, they may not have the ability, the wherewithal, the interest, the willingness to participate in an observational study. So we do have to recognize that cohorts enrolled through observational studies are at some level fundamentally different. But it's also the case, and we've done this, for example, with some of our biomarker work through the CReATe Consortium. While we've enrolled in an observational study a larger population, when we wanted to do biomarker work that we want to have relevance to clinical trial development, we then take the subset of patients from that natural history study who would have met eligibility criteria for a clinical trial had they been enrolling in a clinical trial. So what we call sort of quote unquote trial-like. So that what we're trying to do is avoid that enrichment by focusing on a subset of the population that we've enrolled. So that's one way in which one could, for example, overcome those sorts of biases. Another might be if we think as a generality, C9 carriers account for about 10% of patients coming to an ALS clinic. We could, in a biomarker study, sort of restrict, the criteria for whose samples are used to sort of match that. So I think, again, this is all about the intended use. If you're wanting to, I don't know, develop a biomarker for a C9 gene therapy, well, the biomarker work may need to be done in a C9 population because the biomarker may behave differently in that population. So I think it's about understanding how the biomarker will be used, which we spoke about, and making sure as you then sort of design and conduct that biomarker study that you're paying attention to all the necessary rigor to ensure that it's going to be most relevant to the application that you want to use it for.
And then in the landscape of ALS trials that we've had, there are trials that will bring in specific genetic subtypes, but we don't really see any matching to a drug in any other way. So I feel like we don't have very many predictive markers that are being developed and paired with drugs to decide who might benefit from a drug as we go into trial. So can you talk a little bit about predictive markers, and maybe why haven't we seen more of them? Are we trending towards more of them? Or is it too early in the science to do so?
It's an excellent question. So you raise a type of biomarker I didn't discuss earlier, perhaps intentionally. So we spoke about diagnostic and prognostic and response, and predictive is another type of biomarker. And just to sort of explain, to be sure folks listening get it, because predictive sounds like prognostic. right? When I spoke about prognostic, I said it's predicting something about the future. It's the future course of disease. The notion or the idea behind a predictive biomarker is it would tell us which patient is likely to benefit from which treatment. So let's start with the genetic realm because it sort of illustrates the example well. We probably wouldn't give an SOD1 gene therapy to somebody who doesn't carry an SOD1 mutation. We likely wouldn't give a C9 gene therapy to somebody who doesn't carry a C9 repeat expansion. We sort of use the genetic test in a sense as a predictive biomarker to enrich the population that stands to benefit from the therapeutic. And you're asking the million-dollar question, which is, can we find ways to enrich ALS clinical trials whereby patients enrolled most stand to benefit from the mechanism of action of the drug? So to put a finer point on that, let's say we're studying a drug that targets oxidative stress. Could we find patients in whom oxidative stress seems to be the driving cause of disease or a driving mechanism of disease? Or if we're testing an anti-inflammatory agent, can we identify the subset of people in whom inflammation seems to be driving the disease? The reason we haven't identified these biomarkers yet, maybe this is circular, is that we've not really figured out what to measure that tells us about what those driving forces are. What I think happens in ALS is there's an initial, think about throwing a pebble into the water. There's that initial sort of plop, and then you get these sort of rings that sort of spread out. That in this analogy, right, the stone dropping into the water is the cause. And then the ripples are the downstream biology, what emerges. And it's likely the case that multiple biologies emerge. There's an element of oxidative stress, an element of mitochondrial dysfunction, an element of excitotoxicity, an element of protein misfolding, an element of neuroinflammation. And one of the things we don't know is how much of those are response, reaction to neurodegeneration, and to what extent are they drivers of neurodegeneration. And we don't have great tools to measure those different biological processes in life. People have done work looking at this in postmortem tissue, but it's not clear what that means to people living with the disease or determining eligibility for a clinical trial. So you've touched on what I think is one of the most pressing needs in the biomarker space, is a need for and an urgency for us to develop predictive markers in people with non-genetic ALS that tell us, is there a subset of people likely to respond to this particular therapeutic? We currently don't have those, and I think we have to go after them, but maybe just to say, I also don't know whether they exist. Remember I said earlier that 95 or 97% of all ALS is TDP-43 pathology. Maybe if we're developing a therapeutic that targets that TDP-43 pathology, maybe that's just everybody who doesn't have an SOD1 or a FUS mutation. And so maybe we're sort of making a false predicate here of saying there are different patients sporadic patients who need a different therapeutic. That may be true. I see that as a hypothesis. I just don't know that it's true because we don't yet have the evidence that different pathobiologies are driving disease differently in different people. So that's an open question for me, but definitely one where we need more research.
Thank you so much for going into that. So on that same note, there is a lot of discussion, and more recently in the last few years that I've heard about TDP-43 biomarkers, since it is such a big part of ALS biology across the board. So what do you think have been some of the obstacles in developing those TDP-43 biomarkers?
So maybe just up front, I can say that I'm not the TDP-43 guru, so I'll give you the perspective that I have, and perhaps you'll invite others onto your podcast to talk sort of more about this. I think part of the challenge is that what do we know about the pathobiology of TDP-43? How is it going wrong sort of in people with ALS? And there are two parts to this. One part of this is that in the normal state, TDP lives in the nucleus and it shuttles between the nucleus and the cytoplasm. And it plays an important role in the nucleus in regulating the transcription of many genes. And so it's a regulator of many different cellular processes through that gene regulation. What seems to happen in ALS is that TDP-43 is extruded from the nucleus and tends to aggregate in the cytoplasm. So there are two things going on here. There's a loss of normal function in the nucleus that may be deleterious and having a driving effect on the disease. But it's also the case that the TDP-43 is sort of aggregating, accumulating in the cytoplasm, and that in its own right may be damaging to the cells, to the neurons. And so there are two sort of parts to that. So then you have to ask, well, let's say we're measuring TDP-43 protein in CSF. And those assays have not successfully been developed, but I'm giving a theoretical construct here. What would you expect in normal circumstance? High levels or low levels? I don't know. What would you expect in a response to a therapeutic? Would you expect the levels to go down or go up? I don't know, because I don't know how to tie that back to what's going on in the tissue, in the brain, in the spinal cord. The same is true of extracellular vesicle TDP-43. I'm not sure what to make of an elevated level of that in an exosome or a vesicle. Is that a good thing? The nervous system is clearing unwanted TDP-43? Is it a bad thing? And so without understanding that biological link, I don't know that we know how to interpret sort of purported claims about the impact of an experimental drug on those sort of more speculative biomarkers. So there is another idea we've raised here, and we were talking earlier about the development and discovery and validation. We sort of have to understand the biological plausibility of what it is we're measuring. If I come back to neurofilament, we know this is a structural component of axons, and when axons die, neurofilament is released into blood. And in CSF, so we take sort of a high neurofilament to mean axons are dying, and that's a bad thing. If we can lower neurofilament, axons maybe are dying less quickly. We have to understand something about what the biological interpretation is of whatever we're measuring about TDP-43. This is where I like the idea of this new field that's emerging of these cryptic peptides. This idea that when TDP-43 is lost from the nucleus and it leads to sort of missplicing or a misregulation of splicing, and you generate new peptides or new proteins that didn't exist before, and now we can measure those. I can tie that back to the biology. I can say, if there's a loss of function, I expect those new peptide levels to go up. And if I could have a drug that's targeting putting TDP-43 back into the nucleus, I would expect the levels of that cryptic peptide to go down. And so I don't know that I have that right, but there's a biological plausibility to that that aids the interpretation. Of course, that doesn't undermine the need for the analytic rigor that we discussed early. In fact, it makes it even more important that we do that developmental work to have those robust assays so that we can then ask whether a therapeutic that's targeting restoration of nuclear TDP-43, for example, is having an effect on a biomarker in the way that we would hope and expect it to have.
I think that was very helpful. Yes, in understanding some of the challenges that we faced and also how to tackle some of these issues. So in a trial setting, how would you like to see biomarkers being used differently today? What are we able to do now that hasn't happened historically?
So I'm going to get accused probably of talking too much about NFL, but I think it's a tool that has sort of matured. It's ready for use. We don't know everything about it. There are some caveats, but there are ways in which we can and should, for example, be using neurofilament now in every single clinical trial. So one of the things we spoke about earlier is that it's prognostic. It tells us about the future course of disease. What we didn't talk about is if you do a small, let's say, phase 2 study, 20 patients in one group, 20 in another, or 50 on drug and 50 on placebo, just because of the variability, you could enrich, for example, for slower progressing patients on the active drug and faster progressing patients on the placebo. And if you then saw sort of a slowing or slower disease progression in people on drug, you might attribute that to an effect of the drug. If we have powerful prognostics like NFL that we measure at baseline that can adjust for that known, it's a known source of variability, adjust for it. There may be other sources that we can't measure. Well, we can't address those, but we can use neurofilament today to adjust for that and help us avoid making the mistake of attributing just a group imbalance, which occurred by chance, to the effect of a drug. So I've not yet heard a cogent answer from anybody who disagrees with that view, why we're not doing that in every clinical trial. Phase 1, phase 2, phase 3. We absolutely should. I think we should also be using neurofilament as a readout to tell us, are we affecting the biology, expecting to see a lowering of neurofilament. There are some caveats there. We don't fully understand how neurofilament is cleared. And if your drug, for example, were to affect clearance mechanisms of neurofilament, the drug might be doing a good thing, but you might not see a response in neurofilament because you've affected the disease in a good way, but you've also affected clearance in a bad way. So there are things we don't understand, and there's some caveats to be had, but these are ways in which we should be using neurofilament as a tool that I think is ripe for use in drug development. But I also think we need to be embedding the ability to learn about new biomarkers, which might be in a state of sort of semi-advanced state or in their nascency or new. We should take every clinical trial as an opportunity to do more biomarker development work. One of the keys there is not to overinterpret. If this is the first time that we're looking at a biomarker and it's still got analytic work to be done, let's not make decisions about whether we think the drug is doing something or not. Let's just use this as an opportunity to acquire the samples together with the phenotypic data, do the hard work of discovery, development, validation to get it ready to help us make decisions. So lots and lots of opportunities.
Thank you so much. That was wonderful. Is there anything that you think that we missed in today's great conversation about biomarkers?
I guess the last thing I would say is I think at some level biomarkers are a simple idea, but there's an enormous amount of nuance and complexity that sits below the surface. And I think we don't serve ourselves well as a scientific community, and we don't serve our patient population well as a demography that we're trying to advance therapy development for and look after. We don't serve any of these interests well if we don't engage in good, rigorous science, pay attention to these details, think hard about the nuance, get the interpretation right, and learn from everything we do to make the next experiment better, whether that's developing a biomarker or doing a clinical trial. We have to be sort of a learning system. We're not going to get it right the first time, but we have to collectively learn and get better if we're going to accelerate the pace of therapy development.
Thank you so much. And that's the goal, accelerating therapeutic development. We appreciate your time so much. Thanks for being here today.
Of course, thanks for having me.